Today, I will be talking about microbiome in HIV infection. These are the contents that I will be talking about. HIV or human immunodeficiency virus is a virus that strikes the white blood cells that protects our body from diseases. The most common way of getting infected by HIV is by having unprotected sex. HIV is a virus that stays permanently in the person's body until they pass away. There are two types of HIV. HIV-1 comes from the wild chimpanzees, meaning that if humans keep contact with animals, there's a possibility that the disease will pass on. HIV-2, according to the blood and immunity test, it shows that patients who got infected by HIV-2 showed that the HIV-2 was less harmful than HIV-1. First of all, I would like to talk about the mechanism of HIV. HIV-1 will repeat the genome in many steps, like the virus called retrovirus. Firstly, the infecting HIV binds and enters the cell. Then the HIV uncoated. After that, it undergoes reverse transcription when the RNA is transcripted back to the DNA. After that, the copy of the DNA is inserted into the genome of the cell that is infected. Lastly, the result from expressing the transcription of the latest genomic from the combination of the DNA. Microbiome and immune response. Pseudomonas aeruginosa and Candida albicans are pathogens that cause disease when the host immunity is low, in which most of the patients pass away because of these two pathogens. The good pathogens are bifidobacteria and lactobacilli species. It helps to improve the health of the intestines of the patients. Oral and periodontal microbiomes. Patients who are HIV positive but do not receive ART typically have mouth ulcers or sores, which is the primary indicator of the disease. Among the infected HIV patients, periodontal pathogens are mostly found in their gum. The microbes inside the mouth are also found to be higher with more variety of lactobacillus and the Candida species when compared with the controlled uninfected patients. Candidiasis, an oral lesion caused by yeast, which is Candida, in HIV patients can be lessened by using the antiretroviral therapy. Streptococcus is the main genus that lives in the epithelium at the back of the tongue of the healthy primate, and its main taxa of the oral film are Streptococcus. Microbial species such as Veillonella and P. gingivalis can be commonly detected in people who are infected by HIV, but not with the normal people that have good health. Gut microbiome. The analysis of microbiome by cutting out and removing tissues from the colon help us explain that the HIV-negative patients have less proteobacteria and more firmicute than the HIV-positive patients. The bacteroid is reduced when there is a rise of Prevotella. In the SIV-positive chimpanzees, there are more varieties of bacteria in the intestines. Because white blood cells are destroyed during the HIV infection, the immune system in the body can suffer greatly. Furthermore, there is a change in biodiversity of the microbiome in many parts of the body of HIV positive patients, including the mouth and the intestines. This could be due to bacterial translocation from the gut into the circulatory system. The majority of research indicates that the patients with HIV and primates will, with SIV have a lower diversity of bacterial species in their bodies. Thank you.